Please join me in welcoming to the Distinctive Voices podium, Dr. John Rubenstein.
Thank you, Jennifer, for inviting me, and thank you all for coming in person and virtually. So tonight I'm mainly going to talk about my research, but in the context of how studying a particular one gene and its role in brain development tells us something about the causes of mental disorders such as autism. As you probably know, birth defects and congenital cognitive disorders like autism are pretty common. Maybe 1 in 50 births have some problem in their body or brain. And now we know there are hundreds and probably thousands of different genes that cause these disorders. So getting your hands on trying to understand how they all do it is impossible. You have to break it down into understanding the individual genes and their relative genes and these problems. So tonight what I'm going to do for you is discuss my particular journey in our discovery of the genetics, that is, in genetics discovery that has taught me about how mutation of a specific gene called TBR1 disrupts brain development in mice, and it gives us insights how it does it in humans as well. Mutations in TBR1, in fact, do cause autism and intellectual disability. They're very rare cases in the world, but now tens of them have been discovered. So our research, particularly in mice, provides insights into the brain structures and connections that underlie this human disorder, as I think that the things we understand from the TBR1 mutation have some generalizable principles. Furthermore, understanding the molecular mechanisms of how TBR1 mutants have these defects sheds light on potential therapies, and I'll at the end of the talk tell you about a therapy that we've found for the mice. So about 33 years ago, I started screening for genes that are expressed at high levels in the developing forebrain and at low levels in the adult forebrain, using a method I'm not going to go into. The simple idea that we had at the time was that genes that are expressed at high levels in the developing brain compared to the adult brain, i.e. the adult brain has low levels of them, may be important in controlling brain development. Very simple idea. Using this method, we found many interesting genes. I'll just mention 2 of them. I have a particular reason for doing that. You'll see in about 5 slides. The first one we named TES1. It later became known as DLX2. And DLX2 encodes a gene that makes a protein called a homeodomain transcription factor. Transcription factors are proteins that are molecular switches in our nucleus that turn on and turn off genes on the chromosome. And you may be able to see in this picture, because you can see in this picture, but it's a picture you may not be familiar with, that this gene is expressed in the embryonic basal ganglia. 56 genes later, we found TES56. It became known as TBR1, T-brain-related gene. Unlike DLX, which is expressed in the basal ganglia, TBR1 is expressed in the cerebral cortex. So we were excited because we found genes with very distinct regional expression, cortex versus basal ganglia. Over time, we began to understand that DLX in the basal ganglia regulates the development of GABAergic or inhibitory neurons, whereas TBR1 in the cerebral cortex regulates the development of glutamatergic or excitatory neurons. So here we found genetic pathways for inhibition and another genetic pathway for excitation in the brain. And over the last several decades, these 2 genes became the foundation of my work at UCSF. The naming of the genes is a little bit personal because TES1 is the girl and TBR1 is the boy. Tess is Tess and TBR1 is Thomas Braden Rubenstein. So Anyway, tonight we're going to talk about Thomas Braden Rubenstein, TBR1, and his control of cortical development. I'm going to start by telling you about a mutant called a TBR1 constitutive mutant. That means that this gene is mutated in every cell of the animal, of the mouse, from the time that the fertilization takes place. That's called a constitutive mutant. It's always, the animal always experiences this mutation in every cell over its life. And I'm going to tell you a little bit more about TBR1 expression just to get you in gear on this subject. So TBR1, as I mentioned, it is expressed in the mouse and in the human in cortical excitatory neurons, but not in cortical progenitors. Now, what do I mean by that? So this is a picture. Let's see if I can find a pointer. This is a mouse embryo. This is the head of the mouse. The black area is the cerebral cortex of the mouse. And in the cerebral cortex, the black area are the neurons. The yellow area in the cerebral cortex are the stem cells that make the neurons. TBR1 is the black cells. So TBR1 is not expressed in the stem cells or progenitors that make the neurons. It's only expressed once the neurons are made. Over developmental time, TBR1's expression in the neurons becomes concentrated in the deep layers, like layer 6, shown in this picture. This more superficial layers 5, 4, 3, and 2, have less TBR1, except in the frontal cortex, where layer 5 also has it. We'll get to that later. As the mouse grows up, TBR1 expression is maintained very strongly in layer 6. So the gene turns on at the very beginning of making layer 6 and stays on in layer 6, as far as I know, through the life of the animal. When we made a mutation in TBR1 some time ago, we published that TBR1 regulates the differentiation of layer 6 and the preplate, which is an earlier form of layer 6. Here's our layer 6. Later, with a collaborator, Bin Chen, we provided more information about what TBR1 does. And TBR1, with another gene called FESF2, regulate neuronal identities during cortical development. And so let me just show you a picture of what I mean by that. So this is an immunofluorescent picture of the cerebral cortex, which is that line there. And then this line is shown at higher magnification, and it's stained with an antibody to a protein called CTIP2, which is not important. It's just a marker in this case. And it labels layer 5 and very little layer 6. So there's a lot of red cells in layer 5 and very few in layer 6 in a normal mouse. But if it's a TBR1 mutant mouse, shown down below, you can see that layer 6 now has a ton of red cells compared to the control state. And that is because TBR1 represses layer 5 fate. In the absence of TBR1, layer 6 cells become like layer 5. So it has a powerful role in what we call cell type specification, controlling what cell it's going to become. And in its absence, it switches fate, it changes hats. Layer 6, I'll point out, this zone that's not red, makes neurons which send their axons to the thalamus. Thalamus is a major relay station in the diencephalon. Normally, these axons, which are brown, they're coming from layer 6, and they grow into the thalamus, which is where the red arrow is. They're coming from the cortex and grow to the thalamus, and they came from TBR1 positive cells. In the TBR1 mutant, on the other hand, the axons stop in the basal ganglia and never make it to the thalamus. So TBR1 is essential for making the connection between the cortex and the thalamus, a really important connection. So if we look at a timeline of TBR1 function from embryonic day 10, these mice develop very fast, 20 days in the uterus where they're born. At embryonic day 10, it affects the development of the earliest neurons called Kaharesia cells, which we're not going to talk about. By E12, it's controlling layer 6 fate, embryonic day 12. Between embryonic day 12 and embryonic day 17, it controls the axons that go to the thalamus. So it has these layered effects, the earliest neurons, layer 6 fate, and the axon connections to the thalamus. All of these things go wrong in the constitutive mutant. It's like a house of cards falling down, step by step. Each floor is not constructed properly. In the case of the mice, the animals die around day 3 days after birth. So the bottom line for the TBR1 constitutive mutants is that pyramidal or excitatory neurons in layer 6 are transformed into layer 5-like neurons. That's the big idea for what TBR1 does prenatally in this constitutive mutant. In 2015, Stefan Sanders and Matt State and other co-authors published a paper and found that the TBR1 mutation causes autism. So we got very excited about this, and Matt State became my chairman at psychiatry, and he encouraged me to get back in the fray and study TBR1 some more because of its important role in autism, as well as in intellectual disability. So I returned to TBR1, and my goal was to elucidate phenotypes that may illuminate how TBR1 mutations increase the risk for autism, also called ASD, autism spectrum disorders. Now, in autism spectrum disorders in humans, the individuals have only one bad copy of TBR1. They're heterozygotes. It's very interesting. 2 bad copies is really bad. You'll have a very dysfunctional brain. 1 bad copy, you have autism. So that means we need to look in heterozygotes, which means animals that have only 1 bad copy. And along the way, if we can, let's see if we can find a treatment for these mice that might be translatable to humans. The work we did on TBR1's function was concentrated after the animal was born, postnatally. It was done in cortical layer 6 and a little bit in cortical layer 5. And was largely done by my postdoc, Siavash Fazal Darbandi. And his first paper was published in 2018 on this subject. For this project, we wanted to study the postnatal functions and leave the prenatal functions normal. So we had to build a mouse that's called a conditional mutant. And in a conditional mutant, it's pretty complicated, but you introduce some genetic elements in it, in the gene, that enable it to be cut out at a specific time by a molecular scissors. And this work was done in collaboration with Matt State and Bin Chen. So the molecular scissors we use are called CREs. This is an enzyme that cuts DNA. It's really a DNA scissors. But it only cuts DNA where there are specific signals. So we put 2 signals in the TBR1 gene, flanking essential parts of it, so that when the CRE was present in the nucleus, it cuts those parts out, and TBR1 is destroyed on that chromosome. And so we found some CREs that are expressed in specific layers of the cortex just before birth. Birth is about embryonic day 19, so embryonic day 18.5 is about a half a day before birth. The molecular scissors were controlled by different mechanisms. In order to destroy TBR1 in layer 6, we use this, we call it allele, NTSR1 CRE, which is active in cortical layer 6 beginning at embryonic day 18. So in these mice, TBR1 is totally normal in the whole body and the cortex until embryonic day 18, and then it's cut out only in layer 6. We also did the same operation in layer 5 with another CRE, RBP4 CRE. We'll come back to that later. So for now, we'll talk about what happens if we mutate TBR1 in layer 6 just before birth, giving it a good 18 days of normal development. First thing we did was we isolated RNA from the mutant cortex and then sequenced it, and then this is called a volcano plot, and it's a nice way to look at which genes are upregulated and which genes are downregulated. The downregulated genes on this volcano plot are blue, and the upregulated are red. And you can see that TBR1 is downregulated because we cut the gene out. These are in the homozygotes, so that there's very little TBR1 RNA there. But there's also very little of this gene, and there's a lot of this gene. So let's look what happens in this TBR1 mutant when we remove the gene just before birth. And now we're looking only 4 days later. So just 4 days after losing TBR1, and we're going to look at layer 6 with a marker called TLE4. So this is the staining for TLE4 in a normal mouse. And then this is in the TBR1 conditional mutant, and you can see there's a lot less TLE4 expression. That means removing TBR1 just 4 days later makes these cells lose TLE4 expression, and TLE4 expression is really important for these cells. It is also a major regulator of layer 6 identity. This means that TBR1 is needed not only to induce layer 6, but also to maintain layer 6. You take it away, later the cells lose their identity. This is a marker of layer 5, it's called FESF2. And in the TBR1 homozygote mutants, look at this. Layer 5 is expressing FESF2, and now layer 6 expresses FESF2, whereas normally it does not. So again, TBR1 represses layer 5 fate, and now we learn that TBR1 repression of layer 5 fate continues throughout later development and probably into adult, well, we don't know beyond this age. In the heterozygote, there's a partial phenotype. So this means that individuals that are missing only one copy of TBR1, their layer 6 is confused. It's beginning to think it should be layer 5, and therefore it's going to be abnormal in its development and function. So this result in the heterozygote gives us some hint that what might be happening in the humans who are heterozygote for TBR1, their layer 6 neurons, which are the ones that project to the thalamus, are not normal. And they're thinking they're like layer 5, and it may affect the ability of layer 6 to properly signal and connect with the thalamus. So in this timeline that I introduced you to a few minutes ago, I'm going to add here that gene deletion just before birth, you also get this change of fate from layer 5 to from layer 6 to layer 5 identity. And we don't know how long this goes on throughout life, this requirement of TBR1 for maintenance of cell identity. I'm going to pass this slide because it's too complicated. Just to say that the dendrites of layer 6, they're the antennas of the neurons. The antennas of the layer 6 neurons are usually in a particular location in the cortex. In this mutant, they grow into an abnormal location, and that location is where layer 5 sends its antennas, its dendrites. Now, TBR1 is a transcription factor. That means it's a protein that lives in the nucleus, that binds to DNA, and it binds to DNA at promoters and other regulatory elements. And we wanted to find out where in the genome does TBR1 bind in the developing cortex to understand what its target genes are. What genes is it turning on and turning off? And so we used a technique to do this called chromatin immunoprecipitation, followed by sequencing. And we discovered that the places where TBR1 binds have this particular sequence or combinations of this sequence, and that it binds to both promoters and enhancers of specific genes. And when we looked at some of those specific genes, we found that some of the genes were the same genes that were downregulated in the mutant or upregulated in the mutant. And some of those genes, with the ones with the double stars, are autism genes. So the targets of TBR1 include many autism genes, genes that increase the risk for having autism. We'll come back to this gene here, Wnt7b, because it becomes the star of the later part of the talk. So TBR1 promotes the expression of Wnt7b and TLE4 and these genes, and in the absence of TBR1, the expression of these genes decreases. TBR1 represses these genes, so in the absence of TBR1, these genes are overexpressed, like the FESF2, which is ectopically expressed in layer 6. So a summary of the first part of my talk has these major points. TBR1 is very important in cell identity control. It's required to maintain the identity of layer 6. Layer 6 cells start becoming like layer 5 in the absence of TBR1. Their dendrites take a layer 5 morphology. The combination of RNAs that are expressed in layer 6, that's the transcriptional code, makes it look like layer 5 neurons. And many of the genes that TBR1 regulates are other autism genes. So you can begin to see a pathway where TBR1, itself an autism gene, regulates other autism genes. And we start getting pathways discovered, then we begin to see some convergence of information about what is it genetically that is causing autism. Why are there so many different genes? Well, several of them fall into a particular class. This class I would call the TBR1 regulated class of autism genes. Okay, now we're going to go later in development. We're going to leave the prenatal development. We're going to go into the newborn and adolescent and even adult mouse. And one of the first things that happens after the mouse is born is synapses are formed. Synapses are the connections between one neuron and the other. The axon from neuron A grows and gets close to neuron B, and that axon from A forms a synapse with neuron B. And that's where neurotransmitters are signaling across the synapse to lead to either activation of a neuron with glutamate or inhibition of a neuron with GABA. So here's an example of a green neuron that's forming a synapse with a red neuron. And one can look at these synapses with molecular tools. In this case, we use immunofluorescence to antibodies that are in the components of the synapse to determine if the synapses are there, and if they're there, how many are there? And what we end up doing is looking at the density of synapses, the number of synapses per unit length of dendrite. And we did this in control or normal mice or in TBR1 mutant mice. And this graph shows in the wild type or the normal mouse is red, and the homozygote mutant is blue, and the heterozygote is green. And in the y-axis, we're measuring the densities of synapses. And in the wild type, it has this density. And as we start decreasing from 1 copy gone to 2 copies gone at TBR1, the density of synapses decreases. So TBR1 has a powerful effect on promoting synapses, and in its absence, synapses either don't form or are lost. We collaborate with Vikas Soha's lab, who measured electrophysiology of these neurons to see if it is changed, and he finds when measuring excitatory activity that in the wild type, which has a lot of these little jiggles, that means that the neuron's being tickled. The homozygote mutant is very quiet, consistent with the greatly decreased density of synapses. So in our timeline, let's add synapse density and synaptic activity, which is reduced in the TBR1 mutant. So postnatally, TBR1 now is required for normal numbers of excitatory synapses, and we also found it was also important for the normal number of inhibitory synapses. onto layer 6 pyramidal neurons, onto their antennas, their dendrites. So why are the number of synapses reduced? That's what I'm going to address now. So there's a powerful technique with a super fancy microscope who has this name. I'm not going to go into it. And it takes pictures, and then the computer processes it, and it colors something called dendritic spines, and it makes them blue. So on the dendrite, which is like a tree or an antenna, there are these little nubs that come off of it called the spines. They're like thorns of a thorny bush. And it's on these spines where the excitatory synapses form. Immature spines are long and thin, and mature spines look like a mushroom. So now you get to see some of these spines. So the white structures are the branches of the dendrite, and the little blue guys are mature spines. And this is in a normal layer 6 cell. Now let's look at the density of the blue spines in the TBR1 mutant. And you can see the density of blue is greatly reduced. So this gives us a hint about why the synapses aren't forming. They're not forming because there are very few mature dendritic spines. You need a mature dendritic spine for the synapse to form at high efficiency. This is the quantification. At postnatal day 5, wild type is red, mutant is blue. You can see this diminution of dendritic spine density. Postnatal day 21, this is like a teenage mouse. Control, heterozygote. So one copy of TBR1 taken away, the spine density goes down. 2 copies of TBR1 goes down further. And again, this finding in the heterozygote is very important for thinking about humans because when they have autism from TBR1 mutations, they're heterozygotes. So this would tell us perhaps that in humans, their density of synaptic mature spines is low, and therefore their ability to make synapses is reduced. Okay, so then we went to the next step to try to figure out why are the spines immature. So we went back to our RNA sequencing data on the TBR1 mutant layer 6 cells to look for RNAs that were downregulated or upregulated that could affect maturation of spines. So back to our volcano plot, and we were intrigued by this particular gene called Wnt7b, which is downregulated, it's in the blue. And it's also one of the genes that was directly regulated by TBR1. I showed you about 10 slides earlier. Wnt7b encodes a small protein, it's secreted. And Wnt proteins do different things depending upon the age of the embryo. At this stage of development, they're important in synapse development. So in the normal mouse, Wnt7b is strongly expressed in layer 6. That's what these dark purple dots are. In the heterozygote, it's reduced. In the homozygote, it's really reduced. And then something weird happens up here in layer 2, 3. It gets ectopically expressed. Probably very important. I don't know what it does, and we won't talk about it. We'll forget about it. Okay. Now to figure out whether Wnt7b is indeed part of the reason they don't form, the TBR mutants have a problem forming synapses, we took the cortex out from wild type and TBR1 mutants when they were just newborns. And we plated them in tissue culture cells and let them grow for 24 hours. And on one day after that, we introduced Wnt7b with a virus into these cells. And then we let the cells grow for 2 weeks to see, and then we looked to see whether the synapse numbers were changed as a function of whether we added the Wnt7b or not. Okay, so that's the experiment. In a wild-type animal, you get this density of synapses. In the TBR1 mutant, you get many fewer. This is something I've already showed you before, but here was this recapitulated in this in vitro culture assay. We tried to rescue with a gene called cadherin-8, which was implicated in synapse development in many other cases, it didn't do anything. When we introduced Wnt7b, it caused apparently a complete rescue, which is, I don't know, that's a little bit too much, but it rescued very well. Okay. Now I'm going to turn to doing these synapse assays with the layer 5 mutant. And we like the layer 5 mutant because TBR1 is expressed in the prefrontal cortex in layer 5 and pretty much nowhere else in the cortex. And because the prefrontal cortex is so heavily implicated in thinking and behavior, in psychiatric diseases, we wanted to be able to interrogate it with this layer 5 mutant. So that's what we did. And we collaborated strongly collaborated with Vikas Sohal and Sarah Robinson-Swartz on this part of the project. And this is where we come to our potential therapy for our mice. Okay, so I showed you that TBR1 has decreased synapses. It has decreased Wnt7b. We put Wnt7b back in, and the synapses come back. So then we looked for pharmacological agents that could imitate what Wnt7b is doing, which we could give orally or intraperitoneally to the mouse. And we tried two different small molecules. One is the element lithium. And lithium is an FDA-approved drug for manic depressive disorder. And we tried a more specific molecule called SB216763. Write that down, please. It's from a company from GlaxoSmithKline. No, it isn't. It's from, I can't remember where it's from, actually. But it inhibits the GSK3 beta enzyme, which is important to inhibit with Wnt signaling. Okay. So now what I'm going to show you is just with the lithium chloride. Now we took an animal that was a month old, so this is like a young adult, old teenager, young adult. And we gave an injection of lithium chloride into their intraperitoneal cavity. That's the easiest way to give it in the mouse in a controlled way. Then one day later, we sacrificed the animals, harvested their brains, and counted synapses. Or a month later, we did the same thing. So we looked after 24 hours or after a month, the effect of giving lithium to the TBR1 mutants and their synapse density, or their spine density, or both. Okay, this is to remind you what you already seen. The normal mouse has this density of blue of spines, and the TBR1 mutant has much lower density. So here in the normal, we give it salt water in the belly or lithium in the belly of the animal, and there was no change in the spine density. So lithium did not affect a normal animal in this particular assay. And on the other hand, you may be able to detect that in the mutant, there was an increase in the number of blue spines after the lithium treatment. And when Siavash quantified this in many sections, those were just one section you just saw. If you give saline, the red is the wild type and the blue is the mutant. You can see the mutant has less synapses. That's what we already know. On the other hand, when you give lithium chloride, the mutant now has nearly the same number of blue spines as the wild type. So we were able to, in vivo, rescue spine density in the TBR mutant within 24 hours. It's very fast. It's amazing. With lithium chloride. Then we went on to measure actual synapses. And we found in the saline treated of the mutant, there was no increase in synapses, whereas lithium chloride also rescued synapse density. Spine density was rescued, synapse density was rescued. So in the next paper that Siavash published, in collaboration with Andrew Nelson, Ling Pai, and Kevin Bender, he found that lithium treatment leads to long-term restoration of spine maturation and synaptogenesis and synaptic function in the adult TBR1 mutants. The reason we're doing this is because when a baby is born with a TBR1 mutation, we don't know that. We only know that they have a problem usually about 1.5 years of age to 2 years when autism becomes apparent. So they've already had 2 years of development. Can one intercede after 2 years of abnormal development pharmacologically and improve things? So this is what we're doing in the mouse. Not doing it in the human. We're seeing whether, in this case, an adult mouse can be improved, adult mutant mouse can be improved with lithium. Okay, so let me get this right. All right, so in this experiment, we gave a single dose at 1 month of age, the P30, and then we waited until they were 6 months old. 1 dose at 1 month, wait to 6 months, sacrifice the animal, take out the brain, and count synapses. Here's the saline-treated animal. TBR1 mutant has less synapses. Lithium chloride caused a persistent rescue of synapse density even after 5 months after the treatment. One treatment, wait 5 months, they get rescued. It's just too good to believe. Our collaborators, Andrew Nelson and Kevin Bender, measured the electrophysiology of those neurons to see if they changed, because the synapse density doesn't mean necessarily the electrophysiology will change. Here's an animal that was treated with saline. This is the wild type and the mutant. The mutant has less excitation. That's why the mutant has, its dots are down below here. And with lithium, the wild type and the mutant look indistinguishable, suggesting that the electrophysiology, at least measured by this measurement, which is miniature excitatory postsynaptic currents, forget it. It's just a measurement of electrical excitation of the neurons, is rescued 5 months after giving 1 dose of lithium. I'm sorry, they didn't coordinate properly. This is 1 to 2 months after the dose of lithium. Okay, so summary for this part of the talk. Synapses. This is what's happening postnatally. TBR1 promotes excitatory and inhibitory synapses on layer 5 and layer 6 pyramidal or excitatory neurons. TBR1 promotes Wnt7b expression. It promotes the maturation of dendritic spines and the enhancement of synapse numbers. Spine and synapse defects are rapidly and stably corrected by lithium chloride, even in adulthood. I didn't show this, but the cortical thalamic axon defect was also corrected by lithium chloride. And a quick final section, social behavior in the TBR1 layer 5 mutants and their ability to be rescued by lithium. And this work was done by Mark Turner, Vikta Sohal, and Sivash Darbandi. And this work is not published, and some of it's very preliminary and maybe incorrect, but I'll just show it to you now because it's kind of exciting. Here we're measuring the ability of the TBR1 mutants to interact with other mice. So what you do is you take the TBR1 mutant mouse and put it in a cage with a normal mouse and measure how much time they interact. And you compare it how much time a wild type interacts with another wild type. And the TBR1 mutant has less interaction, a very common finding in many mouse mutants, but this is sort of the mouse equivalent phenotype for decreased social interaction in autism. May not be related at all to the same thing, but this is the assay that we do. And now we do the experiment by giving to the animal saline or lithium chloride and wait 4 weeks. And you can see that the social interaction assay shows a rescue, partial rescue of the TBR1 mutant after lithium. So this is the end of part 3. Social behaviors. TBR1 mutants lose TBR1 in layer 5 in the prefrontal cortex, have social deficits, and the social deficits are rescued by lithium chloride, very preliminary. So conclusions, here's our timeline. Prenatally, it's doing all these things. Postnatally, it's doing all these things. So when you think of an animal or a human who has a mutation in a gene that is expressed in the developing and adult and postnatal brain, there's going to be strata of defects coming from different time points in the life and development of the animal or human. And it's the sum of those defects that leads to the behavior and cognitive problems that that individual has. We don't know which of these deficits in humans are the real problem. It could be all of them, or it could be a subset of them. I hope it's the synapse defects, because I think we maybe can intercede there. Unlike TBR1, most autism genes are broadly expressed throughout the body and the whole central nervous system. So you cannot predict what part of the individual the defect is causing autism. However, TBR1 is so specifically expressed, we can say with certainty about where the problem is. And so the studies of TBR1 are quite useful for showing that the cerebral cortex is the part of the brain, at least in this animal, and I think in probably most cases, that is affected not only in TBR1 mutants, but in autism. And here we show that TBR1 mutants have decreased Wnt signaling that underlies the synaptic defects. And this led us to a potential therapy with lithium chloride or other Wnt agonists that might be helpful in individuals. And I understand there's a group in France who has a cohort of 30 or so human patients with TBR1 mutations who are planning to do a clinical trial with lithium chloride based upon our communication with them. Okay, so that's it. These are all the people that did the work, and I'd like to open up, talk for any questions that you or people in the online might have. Thank you.